Welcome everyone to this video, Thou Shall, Reference Control Commandments for Cytometry. My name is Oliver Eltherington and I am a Senior Technical Specialist at the Melbourne Cytometry platform. In this video, we are going to take a dive into the rules that should be followed when making your single-stained reference or compensation controls, and what can happen if you break them. We will mainly focus on spectral instruments in this video, but these rules are equally applied across conventional instruments as well. Spectral cytometry has rapidly become more mainstream, and, as you probably know, this technique uses multiple detectors that span a wide range of emitted light in order to generate a spectrum of emission after excitation of the particle by the onboard lasers. A multicolour sample, such as this, will contain many different spectrums and the background emission that incorporates the particle autofluorescence plus the signal of the instrument noise. We need to then unmix the multicolour sample into its various constituents. To do this, we need to refer to a signature for each fluorochrome that matches the one in the sample. Now, spectral mixing is not a new technique. In fact, a similar technique is employed in astronomy to determine the composition of planets. It is also used in microscopy to increase the amount of fluorochromes you can use, and it has been used in cytometry before the advent of modern spectral analyzers. To determine what the spectrum of emitted light looks like for a given compound, dye or fluorochrome, you must first establish what the pure signature looks like as a reference. In theory, the more light of different wavelengths that you can collect, the more information about the spectrum that you can use to separate them from one another. With the development of light detection technology, we now have over 64 detectors on board machines, allowing for better spectral mixing. So, here are 4 rules to follow when developing your reference controls. Number 1, the spectrum of emission needs to be unique and identical to the one in your sample. You must ensure that your reference is as bright as possible and should be as bright or brighter than the sample. In order to separate the fluorescent signature from the background, your reference needs to have a negative control that is identical in terms of autofluorescence to the positive. And finally, there must be sufficient events for the positive and negative references. I am not going to cover rule 4 very much, but suffice it to say, the more positive and negative events you have, the more robust the measurement statistics will be. First of all, we will discuss rule number 1, that the spectral signature of the positive reference needs to be identical to the one in the multicolour sample and unique in the panel. I have divided this into 4 topics of discussion. We will discuss using one fluorochrome as a surrogate for another. We will discuss non-tandem versus tandem fluorochromes and their identity. We will talk about minor differences in instrumentation. And finally, we will talk about the eternal choice, beads versus cells. Remember, in spectral cytometry, we are interested in the entire spectrum of emission, and not just the peak emission in conventional cytometry. If you are struggling to get a good single stain control for a fluorescent protein, it can be a good idea to use a different fluorochrome that peaks in the same channel in a conventional cytometer. TD tomato and PE peak in the same detector bandpass pair in this instrument, for example. So, in this instrument, it might be tempting to use PE as a reference for TD tomato. However, if you look at the full spectrum of emission using a 5-laser aurora, you will already see some differences. It is important to look at the normalized signatures once we have removed the autofluorescence for a clear comparison. Let's pause for a moment and just introduce how to normalize the signature. To normalize the PE reference signature, we need to first get the median fluorescence intensity of each detector for the positive and negative sample. We then subtract all the PE negative signature MFIs from the positive. Then, once we have the delta for each detector, we will normalize this delta emission in every detector against the detector with the maximum MFI. In this case, it's yellow green 1. You can plot this on a graph then. This normalized signature allows the software to predict how much spillover into each detector a PE emission will account for. Did you know? When you are in the unmixing wizard in Spectral, you can select Next before pressing Live Unmixing, and Spectral will show you the normalized signature for all the references you have acquired in this experiment. This is a very handy troubleshooting mechanic. Now let's compare the normalized signatures of TD tomato and PE. If we overlay them, you can see where the signatures differ. In fact, if we look at a similarity score, a metric for how similar two signatures are, with 1 being 100% the same and 0 being not showing any similarity in emission at all, you will see that PE and TD tomato are only 0.85 or 85% similar. It is famously stated that Aurora can unmix fluorochromes up to 98% similar. If you use PE as a surrogate for TD tomato in a spectral system, it will overestimate the amount of spillover into signatures that emit in detectors V8 and B4, for example. It will also underestimate the amount of spillover into fluorochromes that emit in yellow-green 3 to 8, for example. It is important to note, you will only see a problem if you are using fluorochromes in your panel that emit in those regions, and of course if you have detectors available for those regions. PE and TD tomato are therefore not identical. To see what happens if we do this, I have an experiment where I am using PE reference to unmix TD tomato from a sample that also contains PE Cy5. Here is how the PE and PE Cy5 normalized spectra look when overlaid. And here is how the TD tomato and PE Cy5 spectra look when overlaid. You can see that TD tomato has a greater emission in the yellow-green 2, 3 and 4 detectors. We will expect to see problems, as mentioned before, because there will be an underestimation as to the amount of light that spills into the yellow-green detectors from TD tomato because we are using a PE reference. So, as a reminder, we are using PE and PE Cy5 references for a sample that has TD tomato and PE Cy5 in it. Initially, I have used PE and PE Cy5 to unmix themselves, and you can see here that it works well. Now, let's look at unmixing TD tomato with PE. As you can see, there is an unmixing issue. This is because there is proportionally more light arriving at the yellow-green detectors than the software is expecting because we've used a PE reference. Clearly, these signatures are different enough that they cannot be used as substitutes for one another. They are, as you can see, so different that you could use them in a panel together. That being said, the amount of spread between the two colours will be large, as shown here. It is also dependent on the signature. Here is BB700 and Percy-PSI 5.5 normalized signatures. Again, two signatures that peak in the same channel and share a similarity of 0.92. However, there is enough difference in their signatures, particularly in the yellow-green region, that when I'm mixing them, the spread is much less. You will also notice that there is a distinct brightness-related difference here. The Percy-PSI 5.5 and BB700 are dimmer than the PE and TD tomato. This absolutely contributes to less spillover spread after unmixing. Combine this with a lack of co-expression and you have the beginnings of some good panel design techniques. My point here, however, is that your reference must be the exact same signature as the one in your sample. This is the basis of all rules in spectral cytometry. There are many ways to break Rule 1. We have just discussed one example of why, for the most part, you can't use surrogate fluorochromes in spectral cytometry. We will move on to the identity of tandem versus non-tandem fluorochromes. As a brief introduction, flow cytometry advancement was initially limited by the discovery of stable fluorochromes that could be easily attached to antibodies and excited by the onboard lasers, such as PE, APC, and FITC, for example. However, with the advent of tandem fluorochromes that utilize FRET, this led to an explosion in fluorochrome tandem development. Briefly, and very simply, tandem fluorochromes work as such. The donor fluorochrome, let's call it PE, is excited by the laser, and some of the energy that would usually be given off as PE emission is transferred as resonance energy to the acceptor fluorochrome instead, in this case Cy7. The acceptor Cy7 then emits photons in its wavelength range. This generates a different spectrum of emission. All of a sudden, this opened up the ability to use larger panels and gather more information. The majority of commercially available antibody fluorochromes are tandems these days. The spectral emission of non-tandem fluorochromes, such as PE, is expected to be 100% similar within a single instrument. So you can use CD4PE from company A as a reference for CD127PE from company B. I have gathered 6 completely different PE references from a single aurora here and put them in the same experiment to look at the similarities. You will see they are all 1 in the similarity indices. Now let's look at a tandem fluorochrome. In this case, Percy P E-fluor 710 in a similarity matrix. You might have guessed from all the foreshadowing, but you can see that there are differences, and in this case they range from about 0.98 to 0.99. In the example of number 1 versus number 3, there are only 98% similar. Spectral flow can unmix up to 98% similar fluorochromes, such as 1 versus 3, admittedly with some significant spread. Let's have a look at why this might occur. The method of generating a tandem fluorochrome is challenging, and between companies and batches the ratio of donor to acceptor fluorochrome emission may vary. It is even harder to control the distance between the donor and acceptor, or even the orientation of the donor to acceptor. Typically, the distance for 50% efficiency of energy transfer in FRET is related to the individual fluorochromes, but it rarely exceeds 3 to 6 nanometers. The orientation of maximum efficiency is also extremely relevant and hard to control. Minor differences in any of these can cause changes in the ratio of emitted light from donor and acceptor. Because of this variability, you should use the exact same tandem from the exact same company, antigen and batch in your reference as you do in your sample. Otherwise, you may expect some unmixing error. Even if you have the same tandem reference in your sample, there is the added issue of tandem breakdown. If, for example, you acquire your reference and then a week later acquire your sample, and there has been some tandem breakdown since the acquisition of the reference, then there will be another mismatch. Tandem breakdown can occur for a number of reasons, which are well explained elsewhere, but it is another issue to look out for. To show you an example of the differences in tandem fluorochromes, I have normalized the signatures of the Percy P E-fluor we saw earlier. Here is number 1, and here is number 3 from the tables. As a reminder, they were 98% similar. Now let's overlay them. You can see some differences between them in the yellow-green 7 to 10 regions and the UV 12 to 15 regions. You will introduce unmixing errors with any other dyes that emit in these regions of difference. They are therefore not identical. What I am getting at here is that the signature of the tandems can vary between company, category number and even lot number. For this reason, we recommend you do not use one tandem as a reference for a different tandem. If you do, then it is essential that you verify that the signature of the fluorochrome in your reference is identical to the one in your sample. It is not always obvious what is or is not a tandem. If you are unsure, check the website of the manufacturer, or we have a not exhaustive list of tandems and non-tandems in the lab and on the Calpendo booking page. The cool kids always use Calpendo. The next example I want to discuss is using a single stained reference acquired on one instrument as a reference for a sample acquired on a different instrument. We have a few 5 laser auroras here in the MCP, so it may be tempting to simply reuse the references from one aurora on another. I have PEs acquired on 3 of our auroras, and then put them in the same experiment to find a similarity matrix. You can see PE1 and PE2 are 1 on the matrix because they are acquired in the same instrument. You can see that each cytometer maintains a 1 similarity for PEs acquired in the same instrument. However, between cytometers there are differences, ranging from 0.97 to 0.99. As PE is not a tandem, we can be sure the signature differences are not because of the tandem issues already discussed. Each cytometer is very slightly different from the other, despite QC and the best engineering. It is practically impossible to make them exact. Here are the normalized spectra of PE2 and PE4 overlaid. You can see minor differences in the area's PE peaks, such as UV9, V8, B4 and yellow-green. You may only see an issue in unmixing if you have a fluorochrome in your panel that overlaps with the area that has an issue, such as BV570. As long as you are acquiring your sample on the same cytometer as you acquired your reference, there should be no issues. Let's have a look at an example. In this experiment, I have acquired PE references on cytometer 1 and 2. I have acquired BV570 reference on cytometer 1. And then I have used these to unmix a sample containing BV570 and PE that was acquired on cytometer 1. So, when using PE as a reference for a sample acquired on the same cytometer, you can see we get good unmixing as the medians line up well. However, when you use the PE reference from a different cytometer, you will see something similar to undercompensation between PE and BV570. Let's remind ourselves of the difference in normalized spectra and add the normalized spectra of BV570 over the top. As you can see, the PE from cytometer 2 is expecting less emission in the BV570 maximum detection area than is actually being detected by cytometer 1. Therefore, by using one reference acquired from a different cytometer for a sample acquired on another instrument, you are breaking rule number 1 and it results in errors. Let's zoom in to have a closer look at the differences between the PEs in the BV570 region. They might be small, but they cause significant errors. So, say it with me, the PEs are not identical. Finally, I want to mention beads versus cells in terms of identity. There are benefits to using cells or beads in your references. It is really dependent on the variety of factors. Brightness of your reference is extremely important, and beads are not always the brightest. Your cell antigen density may be higher than the binding sites on the beads. Additionally, a paper was published this year suggesting that some beads can alter the signature of some fluorochromes. We have even seen this in the MCP. In this example, this is BV570 on cells and beads. It's the same lot number, acquired in the same day and the same instrument. And when overlaid, you can clearly see the differences. We recommend for your first experiment that you bring both beads and cells. Then verify which best unmixes your sample. It might be that you will end up using a mixture of beads and cells for the best unmixing. Rule 1 can be summarised as this. The fluorochrome on your reference must be identical to the fluorochrome on your sample, and we have seen a few examples of how you can break these rules. There are many ways to break Rule 1, as seen here. I'm not going to go into all of them, because that will take a few days, but I have added a couple of honourable mentions to the next few slides. If you don't wash off excess antibody, or you have a carryover from a previous reference, then the reference, in this case PE, will be contaminated. If we look at the spectral profile of the negative in the yellow-green 1 detector, you will see that it's clearly contaminated with another fluorochrome. In this case, it's FITC. After normalization, using the internal negative, you would have a signature that would not match the fluorochrome in the sample. It is even possible to alter the signature of your reference by accidentally getting some small amount of ink into your sample. So be wary of how you mark your wells. We have discussed setting up references in the context of identity. The next rule is as vital. The negative and positive reference need to be well separated, and the positive must be as bright or brighter than the sample. In this section, we will cover why a bright fluorochrome is good, the importance of relevant events, the complications that can arise when using histograms only, and the importance of checking the scale on Aurora. I'm going to steal an idea from photography to illustrate the rule here. If I take a clear image that is well exposed and remove 75% of the brightness, the image is well conserved in terms of colour, for example. This is a bit like your reference. It is relatively easy to predict what a signature would look like when you make it dimmer. However, it is challenging for software to estimate what a brighter signal will look like when using a dim reference. If you have a really dim shot and attempt to brighten it, you will get artifacts, as in this example, where the colours have been artificially enhanced. In cytometry, the software doesn't even attempt to predict. Instead, best case scenario, you will see undercompensation for the fluorochrome in spectra that emit in similar areas, as more photons are associated with that signature than the software is expecting. These are the normalized spectra of BV711 and BUV737. There is considerable overlap, but they are excited maximally off different onboard lasers. In this scenario, we have a reference for BV711 that is as bright as the sample, versus if the reference is dimmer. When the reference is as bright as the sample, you will get good unmixing. However, when you have a dimmer reference than your sample, your sample will be unmixed incorrectly. If we look at the normalized signatures of bright and dim references, they appear to be extremely similar. In fact, Spectralflow's similarity matrix put them at a 1. However, if you zoom in, you can see that there are minor differences in the signatures. There is some loss of signal in the dimmer reference in the secondary peaks. These peaks are in the location of the maximal emission of BUV737. Whilst this does not seem like much, you will see that even a small percentage difference can give a large impact to a bright high MFO. This explains the undercompensation against BUV737. Side note, I want to mention beads again in terms of brightness. Beads are always bright and well separated, aren't they? Well, no, not necessarily. It can be tempting to just run everything on beads all the time, but this is not good practice. We have seen beads are not always as bright as the cells you are unmixing, but we've also seen in a previous rule that the signature is not always the same as the one in the sample. Beads are not the silver bullet of compensation. They certainly have their place, but we recommend again that you use cells and beads and look at how well they unmix your sample. Next, I want to discuss the importance of gating and separation of your positive and negative populations. Here, I have some fixed PBMC that have been stained with a CD4 BV510 antibody. BV510 peaks in V7 in the aurora. If we gate on the brightest possible events, you will see multiple signatures. This is because the autofluorescence of the monocytes here is as bright in the V7 channel as the BV510 positive lymphocytes. If we compare this signature after normalization against the true BV510, you can see it is only 91% similar. It is impossible to gate on mere fluorescence alone here. Therefore, it is essential that you have your sample on scale in scatter so you can change the scatter gate to only include the relevant events, i.e. the lymphocytes. If we do this, and look at the similarity matrix now, you can see that they match. In this next scenario, we have a reference stained with cell trace violet. When we look at the data, there doesn't appear to be a positive population. It might be tempting to just put the gate at the top part of the only peak visible, or even go away and come back with another one. Before you do, however, just remember that histograms are biased towards the majority of the events, and it may be harder to see the positive population if super rare. If we move the gate up to around 10 to the 5, you can see our positive population. Just explore the histograms first. A big problem can occur here if your sample of interest is super rare. Here I have some T cells, and I am expecting some rare signal in PE, but it looks like there is almost none. This is using the auto scaling, which is an issue because it doesn't always show all of the events, and it appears to bias towards the majority. First of all, if I alter the bi-exponential axis to look further below 0, we can see an unmixing error that is quite easily missed in Spectral when using auto axis. So now I can see this, I need to check the brightness of my reference when compared to the sample. It is recommended to use a dot plot to discover the brightest events. So let's check the raw data from the previous scaling issue. So here is the histogram of my reference, and here is the histogram of my sample. They appear to be as bright as one another. However, if I open a dot plot of yellow-green 1 with side scatter, you can see the blue dots are all included in the reference brightness gate that I set. But the red dots are brighter than the reference gate that I set, and very rare, so they don't show up in the histogram. These are the ones that were off the axis in the previous unmixed plot. In this case, the reference is simply not bright enough for these super rare and super bright events. If your target is very rare, then aim to ensure your reference is as bright as possible without being off scale. And consider checking your brightness using a scatter parameter against the peak emission. Because I am on scatter, I can infer that the brightest events are found in my lymphocytes, making this more relevant. Brightness is relatively obvious, but it can be summarized as this. It is a bit like picking out the moon in the night, on a clear sky, through light pollution, or even during the day. You can still tell it is the moon because it's so bright. The brighter your reference, the more detail and information there is about the signature. It also makes it easier to pick out the positive peak in a highly autofluorescent sample, for example. Now we are going to move on to another important rule that we have already alluded to. The negative of your reference absolutely must be identical in terms of autofluorescent signature as the positive sample. In this final section, we will discuss a couple of different ways autofluorescent mismatch between your negative and positive reference can ruin your data. We'll discuss the topic of reusing negative controls, and we'll discuss the challenges of using pan cell markers as a reference. One way to break this rule is to assume that your universal negative has a matched autofluorescence to all of your references. Whilst this can be done, it is only valid if your references all match your negative. It may be necessary, for example, to treat some of your references in a different way to others. Maybe you need to stimulate your cells to upregulate the antigen. Maybe you need to kill some cells to get a good positive for your viability stain. Or maybe you need to use different beads for different antigens. So let's have a look at an example. Here, I have a universal fresh human blood unstained, and I have a fixed unstained sample. You can see the signatures are very different. Let's overlay them to see exactly where. For the most part, the differences are around the mid-UV, the mid-violet and the early blue detectors. I have an experiment here where I am using a universal unstained that is fresh PBMCs, and I have one reference, PE, on a fixed PBMC sample. When these are overlaid, you can see that the autofluorescence of the fixed around the mid-violet and early UV, as well as others, does not match up. Let's have a look at what this looks like when normalizing the signature. Let's have a look at these normalized signatures. On top you can see our mismatched signature, and below you can see the true signature. As mentioned, the worst regions appear to be in the early to mid-violet and the early UV. If matched, we would expect the peaks in the areas of emission and flat lines where there is no emission, or peaks in planes as I like to say. If we use this as our reference, we would expect there to be an overestimation of the contribution of PE into the early violet region, and thus it will apply more compensation than is needed against fluorochromes also emitting in these regions. As a side note, Cytec Cloud is a great resource on the Cytec website that is free to use, and you can compare your normalized signatures against the reference one. The link will be at the end of this talk. To demonstrate this issue, I have made an experiment here containing PE, BV480 and FITC. BV480's peak emission is in the early violets and UVs, where we see the mismatch. You will see that FITC peaks around the blue 2 detectors that are minimally affected by the difference in normalized PE signature. So, as we can see here, the software is expecting more photons to arrive in the detectors associated with BV480 because of the incorporation of the autofluorescence into the signal. Therefore, the sample is overcompensated out of the spillover channels to degrees related to the amount of mismatch, i.e., the autofluorescent mismatch was most prominent in the violet channels, thus BV480 is most affected. Now, let's look at what happens when the unstained sample is brighter in terms of autofluorescence than the single stained reference. Here is the autofluorescence signature of granulocytes, and here is the autofluorescence signature of lymphocytes. Here are the granulocytes again, and here is lymphocytes stained with BUV496. It is quite difficult to see the differences in the autofluorescence in a stained sample, especially when overlaid. So let's look at the normalized signatures to see the differences. So, as before, we have normalized the signature of BUV496 using the brighter granulocyte as a negative. There are some minimal negatives here in the yellow-greens, the late UVs and the mid-blues. Let's compare this to a true BUV496 with a matched autofluorescent negative. So, we might expect some unmixing issues in fluors that emit in these channels, such as yellow-green 1 to 5. Let's say PE, for example. Here is the normalized spectra of PE. You can see that it overlaps with the area of interest. You can see that the BUV496 in the unmatched sample is exhibiting some undercompensation. It is quite little, however, even a small amount of error created can be exacerbated in larger panels when combined with other errors, or when looking at secondary or tertiary antigens, or when the fluorochrome is very bright. This section can be summarized as follows. Whatever you do to the positive reference must be done to the negative to match it, minus, of course, the stain itself. Right, on to the next potential issue. Reusing references is actively encouraged in the Aurora instruments, and as long as they follow the rules already laid out, there should not be too many issues. However, if you decide to add a new reference to a current panel, or update a reference, you should include an unstained control that is acquired on the same day. The reason for this is because the unstained control signal is not just made up of the intrinsic autofluorescence of the particle that is influenced by metabolism or fixation, or the cell type, for example, but there is also a component of the unstained signal that is derived from the instrument noise, be it optical, electrical, or any other source of noise. Just to visualize the point, a few months ago we noticed a fault in one of our auroras that has since been rectified. At 9 AM we ran QC and it passed no problem. We also ran rainbow 8 peak beads. We then repeated this a few hours later. As you can see, in the negative rainbow peak on the left, there is an increase in the low-end signal noise in the blue detectors. If we overlay the positive peaks on the right, there appears to be no problem. This, incidentally, is a flaw of most cytometer tracking or QC beads, because the symptom is often not visible in the brighter peaks. This is one of the reasons we perform extra checks as standard to ensure that we catch any problems earlier. Anyway, if you acquired a reference at 4 PM and used a negative from 9 AM, your normalized signatures would have differences, resulting in unmixing errors. This is an extreme example, but even day-to-day variation is enough to introduce differences into signature. If, however, you acquire the unstained and the stained around 4 PM, the electronic noise will be incorporated into the unstained signal and removed from the normalized signature, resulting in correct unmixing. This is ultimately why you should not reuse unstained controls. Finally, I want to talk about pan-cell markers, i.e. CD45. The issue you will come across will be that there is often no matched negative within the reference control tube. For example, here is CD45 on FITC on PBMCs. If I gate on the CD45 positive peak on the histogram, you will see that all of the cells are CD45 positive. The negative is simply debris, which, if used, will generate this normalized signature. When compared to the correct autofluorescent matched FITC, you can see the issues. For example, the CD45 reference has more signal associated with it in the UV, the violet, the yellow-green, and the red. As shown previously, this will cause unmixing errors with dyes that emit in the same regions. It is essential then to bring an unstained cell sample so that you can use this as a negative for a pan-cell marker. It is also essential your scatter parameters are set correctly so you can gate on the correct population. Of course, you need to know about a bit about the biology too. When you do this and you normalize the spectra, you can see that the extra signal in the UV, violet, yellow-green, and red has gone. If you follow these general rules, you should greatly limit any chances of unmixing errors. Now, that's a wrap. Thank you very much for watching, and watch out for our Spectral troubleshooting video. Here are some links to some extra resources, and I can't stress enough the usefulness of Cytec Cloud when checking normalized signatures. Happy flowing!